Hello, very good afternoon to all of you. I hope that you all are doing well. And today we are here to discuss the last section. I hope I'm audible to all of you. Clearly audible to everyone there.
Yes, sir.
Yes,
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 yes.
sir.
Great, amazing. Okay, so today we are here to discuss the last part of the first section of microbiome, which speaks about the flora of stomach, small intestine, and large intestine. So that's what we are basically going to deal with. And the last section over here speaks about the factors which shape the kind of flora that you have in your gastrointestinal tract, which we have spoken earlier in brief. But we will also see some more details of the kind of work that has been done to prove how our lifestyle, how the mode of delivery, how the antibiotics that we consume have a great influence on the kind of flora that we have in our GI tract. So to begin with, it's very important for all of us to understand that the very first contact with the microorganisms, where it would decide what kind of flora we would possess, is the mode of delivery. Okay, we have spoken earlier about this, that whether it's a normal delivery or whether it is a C-section, whether it's a cesarean, on the basis of that, the kind of organisms that are going to colonize the baby is going to be very different, because the organisms that are present in the vaginal tract and the organisms that uh, coat the baby when it passes through the vaginal tract during the normal process of delivery is way different than the kind of flora that you will find on a baby who is born by C-section, by cesarean. Because under these circumstances, you will see that the baby would have maximum of organisms which come from the hands of people who are handling it for the first time, like the medical personnel, the doctors, the nurses, because the baby is not passing through the vaginal tract. You are surgically removing the baby out of the womb of the mother. So the kind of organisms that they come in contact with are very, very different. And that has a huge influence on the kind of community of microorganisms that the baby is going to have. The second is the feeding of the newborn. I have told you that when it comes to the internal colonization of the baby, the colostrum has a huge significance. Colostrum is nothing but the mother's milk, okay, which the baby is given by the process of breastfeeding. And the kind of organisms that are present in the colostrum have a huge significance in providing a lot of resistance to the baby, because it carries antibodies. And you know that in the initial stages of its life, in the initial months of its life, the baby doesn't have its own immune system. So the antibodies present in this colostrum is going to be very, very significant for the baby to fight any kind of infectious agents that it may come in contact with in its initial months after birth. But we need to understand that along with those antibodies, there are obviously going to be microorganisms from the genera of Lactobacillus and also specifically Bifidobacterium, which is the first kind of organism that the baby's gut is going to have. Okay? And these organisms are very, very crucial because they have a significant role to play in not just providing protection against any kind of pathogenic organisms that may colonize the gut, but they also have a very, very significant role in building up the immunity, the immune system of the baby. Obviously, when the baby switches from the breastfeeding to the weaning process means where you start giving it the food that we all normally consume, there is going to be a shift in the kind of microflora. There is going to be a shift in the kind of community that you will find in the gut of these babies. Okay, and obviously that also has a significant role in a way in further diversifying the kind of immunity that this baby is going to possess. Timely exposure to antibiotics, to drugs, whenever you are unwell, whenever you are under some kind of treatment. These antibiotics also could have an effect on the kind of community that you possess inside your GI tract, but there are some surprising facts that we will be seeing today when it comes to the influence of antibiotics on the gut microbiome. Other than that, your genetic factors. See, please understand that genetically, we all are very, very different from each other. The kind of resistance that we possess, the intensity of immunological response that each one of us is going to cast is going to be very different. And all this is because genetically, we all are very, very different from each other. So the kind of immune system that I have may be too weak as compared to the immune system that probably Aman has or Apurva has or Akshata has, because we all are genetically different individuals. So how does my system react when I have a specific kind of community in my GI tract also matters a lot. Because maybe Akshata has some kind of an organism which is an opportunistic pathogen, but her system manages it well, keeps it under control, so that organism's presence doesn't cause much harm. But maybe if my immune system is weak, my system is not able to fight or keep up with the opportunistic pathogens, and that particular organism being a part of my gut microflora, being a part of my gut microcommunity is going to cause a trouble for me. So even though I and maybe Akshata possess the same set of organisms, but the way my body responds to that community and the way her body responds to that community is going to be very, very different from each other. Also, please understand that some kind of infections, disorders, diseases are going to have an influence on the kind of community that you possess. And basically, most of the time, this is in case of severe infectious agents, wherein your gastrointestinal tract is involved, like suffering from typhoid, suffering from cholera, has been found to have a great influence on the kind of microbial community that you may possess after the infection is done. So which means that before the infection, you had a specific type of a community of microorganisms in the gut. But eventually, post-infection, the influence is very, very evident, and you can see that the community is shifting gradually. Also, surgical interventions may disturb the community, which means that there was a surgery that had to be performed, which is in the abdominal region. And this surgery could introduce some new set of microorganisms which were completely unique, which were completely new for your gut system. But that microorganism has started flourishing in your gut and now has started residing there. So that could also be responsible for shifting the kind of community that you possess in the gut. So I hope how all these factors are in general causing an influence on the microbial community is quite clear to all of you. Yes? Does it make sense
Yes,
to
sir.
you what I'm speaking here?
Yes.
Yes, sir.
Yes, sir.
Yes.
Yes, sir.
Okay, great. So just let me know if I'm going too fast, or if you want me to repeat something, please feel free to stop me and tell that, sir, let's have a look over it again if possible. Okay, so we can surely do that. Okay, so the first thing that we're going to have a look at is the microflora of the neonates and the children, which means that the newly born fetus. Okay, so from there to a few years time span, what kind of changes am I actually observing in the fetus? This is quite significant. So I have told you that it's an assumption. Okay, I am again repeating in inverted commas. It is an assumption that the human fetus is sterile before birth. So I've been telling you that people have had these notions for years together that the uterus is sterile. Okay, so the baby that's going to be in the womb of the mother is ideally going to be sterile and free of microorganisms. But I had told you previously also that there are several researchers who are contradicting this fact. According to them, there is kind of interaction between the fetus and some microorganisms which have been harbored by the mother's system for a very long period of time. So the first exposure of the baby with the microorganisms begins in the uterus itself. Okay, but still this is something which has a lack of a lot of scientific support, because of which still people are working out to come up with concrete evidences to put this as a well-known fact. So till now, books have been referring to the same thing, that the baby in the uterus is considered to be sterile unless and until the mother is suffering from some kind of an infection, and that's a uterine infection where pathogens have reached the uterus. So until and unless that happens, the baby is going to be considered to be sterile. And the first contact with the microorganisms takes place during the process of delivery, okay, where the vaginally born children, when they pass through the vaginal tract, they are going to come in contact with the flora that's going to be present in the vagina, and also some flora which is present in the fecal matter of the mother, okay, which is going to coat up the baby's body, and that's the first coat of microorganisms that the baby is going to acquire. But the children who are born with cesarean section, they obviously are going to get the microorganisms which are present on the skin of the mother, and at the same time, obviously, microorganisms which are harbored by the medical personnel, like the nurses and the doctors, which handle the newborn baby as it is born. Okay, so it has been proved by research that babies that are delivered by C-section and not by normal delivery, they show a delayed or reduced or absent colonization with important groups of bacteria, okay, which includes organisms from bifidobacterium and even bacteroids genera. So it means that there is a possibility that they won't have these normal flora at all, or it may take some amount of time than normal, which means it's going to be a delayed process of them getting these organisms in them, or their numbers, even if they are present, are not as adequate as it would be in case of a normally born child through the normal process of delivery. Okay. Instead, they could have organisms like Clostridium difficile group, which is an obligate anaerobe, and also there could be organisms from Staphylococci group. Okay, so completely a different set of organisms is what they would possess as compared to the children who are born through the normal process of delivery. Now, a lot of 16S rRNA sequencing has been performed, okay, and this has been done across the globe because there have been several research groups which have been closely working together, wherein they have been trying to understand the microflora of the newborn individuals and of young children, and to see that what kind of changes takes place with time in their skin, in their oral cavity, and in their gastrointestinal tract-based microflora. Okay, so the close analysis using molecular biology techniques, using 16S rRNA sequencing, they have found that the microflora which is present on the vaginally delivered baby's skin, it resembles to a great extent to that of the microorganisms which are present in the vagina of the mother. So obviously this kind of a study would require meticulous sampling from the vagina of the mother and sampling from the baby to compare the kind of organisms that they possess. Okay, and this cannot be a ikka dukka case. No, you have to have a large group of study. So research communities, researchers will have to, in a way, have association with pediatric wards in different hospitals, in gynecology-based hospitals, wherein they have tie-ups with these institutions that we would want to take samples of the newly born baby as well as of the mother, and we would want to do this kind of a study. Obviously, this requires permits. This requires permits not just from the hospital, but also from the volunteers who are participating in this process. So in this case, obviously, baby is not going to be able to give you a permit, but the permit of the mother, the permit of the parents, both the parents, is going to be crucial for taking samples and carrying out this kind of an analysis. Okay. Also, there have been classical culture-based studies wherein they have seen that after a weaning process. Now, what is weaning? Weaning means that the baby has started consuming food that, like, we all consume. So once you start giving solid food to the baby, you see that the kind of microorganisms that they have in their gut, they have in their colon, they have in their feces is all matching to that of the other. which means that they have started having a community shift from what they had earlier and what they have now after starting consuming the solid food material. And once they get that flora remains quite stable and constant, and very little day-to-day variations will take place until and unless you are consuming some kind of food which is contaminated with completely different set of microorganisms. Now, please understand that this study becomes very significant when you see that the kind of flora in adults in one geographical location differs a lot from the adults living in a completely different geographical location. So you cannot expect an Asian gut microbiome to match the European gut microbiome or the US gut microbiome. Surely, there might be organisms which are similar, which are same, but you cannot expect the same community to be present. Because according to the geographical conditions, the kind of microorganisms that are found in those locations is also bound to vary. So we cannot expect that in the entire human species or in the whole mankind, I am going to have the same kind of gut flora. ऐसे बिल्कुल भी नहीं होता. Within the Asian countries also, you will see that there are variations. Within a specific country also, you might find variations. Now, why is this so? This is because the geographical conditions are different, the climatic conditions are different, the kind of diet that we have is different, and even the lifestyle habits may differ to a great extent. So all these would add to variations, but within us, let us say that we all are in Mumbai, so within us, the variations might be present, but to a less extent as compared to comparing our gut microflora with, say, China's individual gut microflora or Japan individual gut microflora. Okay, fine. So also there have been a lot of, different kinds of techniques that people perform. Like some people stick to culture-based analysis. Some say that no, culture-based analysis is not correct because you don't get a correct representative set of organisms, because we know that we are yet not able to cultivate all types of microorganisms in the lab. That is one of our disadvantages or limitations to a great extent कि हम सब type की microorganisms को lab में grow नहीं कर पाते हैं. तो कुछ communities हैं, कुछ scientific communities हैं जो ये कहते हैं कि नहीं भाई हमको 16s rRNA ही करना चाहिए. तो कुछ scientific community 16s rRNA को भी नहीं मानता है. They say that अरे यार ये टेक्निक भी लिमिटेड है, क्योंकि यहाँ पर 16s rRNA करने के लिए आपको पहले कंजर्व्ड सीक्वेंसेस के बारे में पता होना चाहिए इन एवरी जनरा. अगर वो आपके पास नहीं है तो आइडेंटिफिकेशन का जो प्रोसेस है वो कंप्लीट नहीं हो सकता. देयर आर चांसेस दैट यू मिस आउट फ्यू ऑर्गेनिजम्स इन 16s rRNA also. So people say that you go for metagenomic analysis, wherein you ensure that you are not missing out any single microorganism in the process. But no doubt that is really very expensive, and you need that kind of a funding and that kind of an infrastructure in your lab to do those kind of studies persistently in the laboratory, right? So obviously, when you do a culture-based analysis, a 16s rRNA analysis, and metagenome analysis on the same sample, you will see a lot of difference. You will see that what you got on culture-based technique was very less flora. In 16s, it phenomenally increased, and in metagenome, it increased much more times as what it was in case of 16s rRNA as well. So you will see that people report differences in number of organisms found, especially when they compare the flora in baby, adolescent or individuals and adult individuals. So ये जो stage है ना, adolescence, इस पे अब साइंटिफिक कम्युनिटी का ध्यान बहुत बढ़ रहा है, क्योंकि adolescence हमको पता है कि is a stage wherein there are lot of changes that are taking place in the body in terms of physiological and hormonal changes. So people are more interested to understand, does the kind of microflora that you possess during your adolescence age, does it bring a huge difference? That is a very interesting study, because if you are able to link that, you will be able to understand that what kind of a community would help me for a better development during the stage of adolescence. because there are a lot of problems that individuals undergo during their adolescent stage, okay, in terms of developmental delays. You would have seen that there are several people who have developmental delays. Their secondary sexual characteristics are delayed, not because they are genetically predisposed to it, not because they have disorders, but there could be something which interferes in their process of development. Okay, so is microflora related to that in any way? That would be a very interesting study to perform, right? Okay, so one of the most important, thing that we all should note is what kind of diet is fed to the newborns during their first months of life. So you would have seen that जब आपके घर में बड़े बूढ़े होते हैं, ग्रैंड पेरेंट्स होते हैं, उनको पता होता है कि जब हम ब्रेस्ट फीडिंग से we are shifting to a diet, a solid food diet, what kind of food stuff should we give? ओके, और वो स्पेसिफिक फूड स्टफ्स होते हैं जो वो बनाते हैं अपने ग्रैंड चिल्ड्रन के लिए, क्योंकि उनको पता होता है कि ये काइंड ऑफ फूड स्टफ बच्चा जो है पचा सकता है, डाइजेस्ट कर सकता है, और कुछ काइंड ऑफ फूड स्टफ्स है जो डाइजेस्ट करने के लिए अभी भी उसका एबिलिटी डेवलप नहीं हुआ है। तो ऐसे फूड स्टफ देने पर ब्लोटिंग के चांसेस होते हैं, गैस डेवलप होने के चांसेस होते हैं, इनडाइजेशन होने के चांसेस होते हैं। सो दे हैव स्पेसिफिक काइंड ऑफ फूड स्टफ दैट यू वुड गिव, right? And at the same time, if you see what are the products available in the market, you will see that even in the market, there are commercial products which are considered as approved solid food products that could be given to the baby when it is shifting from that stage of breastfeeding to solid food. I'm sure that most of you would have observed this, right? But घर में बड़े बूढ़े होंगे तो वो बोलते हैं ये मार्केट का प्रोडक्ट छोड़ो, हम जो बता रहे हैं वो करो. And that works also because obviously they have learned that from their experience, from their elders, and it has been following in our generation for God knows how many generations altogether, right? Now, breastfed children, no doubt they are exposed to microorganisms present in the breast milk, but they are also exposed to the bacteria that is present on the skin of the mother. Because please understand that during breastfeeding, the baby is also coming in contact with the skin of the mother. So the overall hygiene during that stage of breastfeeding matters a lot. आपने ये भी देखा होगा अगर कभी आपने closely किसी cousin के या घर में किसी भी family member के pregnancy को देखा है, तो आपने ये notice किया होगा कि काफी बार ये कहा जाता है कि if the mother is unwell, she should not breastfeed the child. Okay, because that could affect the child also, right? So the complete health status of the mother during that stage becomes very, very significant, and you have to be careful, right? Because that could affect the baby in a disastrous way. Okay, now in the Western culture there is a lot of change when it comes to taking care of children, because there is a lot of bottle feeding that is done over breastfeeding. Okay, and that's the reason why WHO also has been taking these efforts in several countries in different ways to make people understand that colostrum has significance. You cannot refute the significance of colostrum, but people still, there are people who stick to the bottle feeding practices. Okay, but that also has to be taken care of, because it has been seen that bottle feeding practices could affect the kind of microbiome that the baby develops. Okay, they may end up getting only very few species which are beneficial, and they may end up getting all unnecessary species of organisms that comes from the bottle if it's not maintained clean and sterile. So we need to understand that there is some significance of the natural process, and that cannot be replaced with these kind of techniques. Okay, and it has been proven that bottle-fed infants, they are more colonized with organisms like E. coli, Clostridium difficile, bacteroids, okay, and lactobacilli, specific species of lactobacilli, which may not be really very significant and beneficial as compared to the breastfed children. So breastfed children get more different varieties of lactobacilli and bifidobacterium, which helps the baby, which has potentially beneficial characteristics, which the bottle-fed children may not get. Okay, so you need to understand that it becomes very, very important that we follow the natural process. Now, in some cases in breastfed children, it has been found that they get fecal bacteria like bifidobacteria, but those strains may not be really beneficial in a big way. Okay, so if you want to get beneficial strains of bifidobacteria, beneficial strains of lactobacilli, breastfeeding has to be practiced. Okay, so these kind of studies have added significantly to the understanding that why breastfeeding is considered to be essential. Now, whatever you may do, you do breastfeeding, you do bottle feeding, it is seen that once the baby starts having solid food, once the weaning process begins, whatever kind of microflora the bacha had in the gut, that's going to change. And everyone is ultimately going to get the adult type of microflora, which we all acquire from the kind of food that we have, from the kind of diet that we have. Okay. Now, your question here should be, फिर इतना मेहनत करने का ही काय को? Okay, then it is better, no, you do either breastfeeding or you do bottle feeding. How does it matter? Please understand that those microorganisms have a significance at that stage to help you develop your immune system. So you cannot refute its significance at that stage. After that, everyone's microflora is going to shift. That is okay. But at that stage, these microorganisms are essential, and that's why breastfeeding becomes essential, even though this flora might shift at a later point of time. Right? And that's why if you see the last point on the slide, they say that how tempting it is, which means how interesting it's going to be to speculate, to assume, to find out and explore that the differences in the gut microbiome, how is it going to impact the early stages of development? How it is going to have an impact on the immune system development? And how it is going to impact the overall well-being of the baby in the initial months after birth. So that kind of a study will again involve a lot of omics approach, because you will have to see what kind of metabolome do you observe in the gut in those stages of breastfeeding wherein they have those specific beneficial strains. So what kind of metabolites are produced by the microorganisms there, and how are those metabolites having an importance in developing my immune system, in helping me develop my systems further, is going to be very, very interesting. Coming to the adults. So before we go to the adults, do you have any specific questions till this point? मैं बहुत बकवास कर रहा हूँ, मुझे कुछ response नहीं दिख रहा है. So are you understanding the points here? And if you have any specific questions to ask, please shoot. Hello? Any questions to ask? Yes, no, you can type it, say.
No, sir.
No, sir.
We are understanding, sir.
Okay. Thank you. So let's go ahead then without wasting time. So we are now talking about the microflora and the others. Okay. Now, please understand that for every individual, जैसे हर एक का फिंगरप्रिंट अलग होता है, वैसे हर एक का माइक्रोबियल प्रिंट भी अलग अलग होता है. Okay, and that is unique for them. That's going to be specific for them, and it's going to be relatively stable over time until and unless there are some devastating situations which creates a complete imbalance, and obviously that's going to be considered to be abnormal. But under normal situations, this community is considered to be very stable. Okay, no doubt that there could be significant additions and deletions in the composition, which may be related to the kind of diets that you have, to any diseases or disorders that you suffer from, to traveling, because as you go to different geographical locations, the kind of climatic conditions around you, environmental conditions around you, the kind of diets that you would have is going to change. Okay, so obviously it's going to have an impact eventually on the kind of microbiome that you possess. To just give you an example to explain this to you, I'm sure that you would have seen that several people who come from abroad to our country, you would have seen that वो बिस्लरी का बॉटल लेके घूमते हैं. देखा है क्या तुम लोगों ने कभी? मतलब बहुत funny लगता है, but wherever they go, they will carry small Bisleri bottles with them, and they avoid drinking water in and around us. Have you seen that? Have you
Yes,
observed
sir.
that?
Yes,
Yes, sir.
sir.
Yes.
Do you know
Yes,
why?
sir.
Have you thought why? Have you thought why?
Sir, because water can harbor many type of organism. For
Okay,
it may be contaminated also, so they avoid to drink our water. Like,
okay, but you yourself are consuming that water, right? Many a times you would have seen this also. Matlab, to just give a hint more towards what I'm trying to say, you would have seen that तुम भी वही चीज खाते हो या पीते हो और वो भी वही चीज उसी वक्त खाते हैं या पीते हैं, आपको कुछ नहीं होता, वो बीमार पड़ते हैं, देखा है कभी?
yes, sir.
Yeah. So have you given a thought about it? Why does this happen? Anyone?
Difference in immune system.
Okay. What else? Differences in immune system.
Sir, we are exposed to microorganism, like
Okay, we are
uh,
exposed to microorganisms. Okay, this could
different
be put
kind.
it
Yeah.
in. Yeah. Okay. Okay. Anything else that anyone can think of?
So our microbial community is very well adapted and developed.
Exactly. Yes. So please understand that the kind of microorganisms that they possess and the kind of microorganisms that we possess is going to be way different. Okay. And you need to understand that we have continuous exposure to microorganisms, because of which, even in terms of immunity, we are one step ahead as compared to them, because of the kind of exposure that we have. So even if the water had something which was minor, okay, which is not a potent pathogen, it's a minor pathogen, it may not affect me because my system is habituated to it. But their system finds it to be a completely different organism and they don't know the way to deal with it. Okay? So under such circumstances, they fall sick. Now, let me tell you another interesting fact, that if you go to a foreign country and, you stay there for a long period of time, let's say for 15 years, 20 years, and you're coming back. And let me tell you that you also will face this problem. Because eventually, as you settle there and you got adapted to the kind of diet that they have, there might occur significant changes in the microflora that you possess. So people who are true residents of Mumbai maybe, and they go and stay back there and they come here after years together, they may face problems initially. Eventually they may get adapted, no doubt about it. But there are going to be initial glitches that they will all have to face. Okay, fine. Now, as far as antibiotic treatment is concerned, uh, no doubt that people have always been thinking that antibiotics will bring about huge change in the microorganisms or the microflora that we possess. Yes, but let me tell you that don't expect the entire community to change. It's not that if you had 1,000 species in your gut, 999 out of it is going to die. No, it doesn't work like that. Yes, significant number of organisms may die. Okay, there might be around 40% of the flora which dies, and that too brings a significant effect, because whatever role they had to play there now, there's no one to play. So there is going to be a manifestation of this effect in you, which you show in the form of acidity bouts or some other kind of side effect. So that's going to happen. But don't expect the entire microbial community to change. That doesn't happen. Okay? Even after the recovery period, if you compare the kind of flora that they possessed before and after the treatment, you will see that more or less it remains stable. It doesn't change drastically कि हाँ कुछ तो मर गया, कुछ तो और आ गया and मेरा community पूरा change हो चुका है. ऐसे नहीं होता. कुछ deductions होंगे, कुछ additions होंगे, but it's not a completely brand new microflora that I'm going to possess in my gut. That doesn't work like that. Yes, if you are on a drug or an antibiotic which continues for months together, for years together, then there are chances that I can point out significant differences that okay ऐसा हुआ करता था माइक्रोबायोम, लेकिन इतने सालों तक कंटिन्यूसली वो ड्रग लेने की वजह से या वो एंटीबायोटिक लेने की वजह से अभी मेरा जो माइक्रोबायोम है वो ये हो चुका है. So that can be mapped, that difference can be mapped, but when you are continuously taking that antibiotic for a longer period of time, right? So that makes you understand the difference of, you know, what we think that antibiotic does to my gut microbiome and what actually happens. Okay, now please understand that as you age, the gut microbiome is going to change. Okay, so in elderly people you will see that Enterobacteriaceae, Bacteroidetes group के जो organisms है उनका नंबर बढ़ता है और Bifidobacteria जो potentially beneficial strains है Bifidobacteria के उनका नंबर कम होने लगता है, जिसकी वजह से आपको digestion related issues, acidity related issues aged people में काफी commonly होते हुए नजर आएगा, जहां पर वो काफी commonly होता है. They say that अभी मुझे ये पचता नहीं है, ये मुझे digest नहीं होता है. Fibrous food consume करने पर they have diarrhea, they have bouts of diarrhea. तो पालक या मेथी ये चीजें वो ज्यादा आसानी से पचा नहीं पाते हैं as they grow old, because the kind of microflora that they possess in their gut, which used to help them to break these fibrous foods down effectively, it has started changing as you age. Okay, so this is also because there are differences in the kind of food that you consume when you start aging. The amount of food that you consume starts changing, the type of food that you consume starts changing. Gut motility जो है वो डिक्रीज हो जाता है, क्योंकि जितने एक्टिवली पेरिस्टाल्सिस होता था उतने एक्टिवली पेरिस्टाल्सिस अब नहीं हो पाता, तो जो रिटेंशन टाइम है फूड का वो चेंज होने लगता है. सारे जो फिजियोलॉजिकल कैरेक्टरिस्टिक्स है वो चेंज होने लगते हैं, और बुढ़ापे के साथ बीमारियां आती है तो अलग अलग टाइप की मेडिसिन तो चलते ही रहते हैं. ओके, एंड मूवमेंट ऑफ द बॉडी इज ऑल्सो लेस. ओके, ज्यादातर सिडेंटरी लाइफस्टाइल हो जाता है, मोटिलिटी कम हो जाता है. ओके, चलना फिरना कम हो जाता है, तो ऑब्वियसली दैट्स गोइंग टू हैव सम काइंड ऑफ एन इफेक्ट ऑन द टाइप ऑफ microorganisms that you possess. And along with this, researchers have established a link now that obesity, insulin resistance based diabetes and type 2 diabetes, this is connected to your intestinal microflora. You are going to observe that there is reduced proportions of beneficial clostridial species. Clostridia is a part of firmicutes. So there is a reduction in those beneficial species of Clostridia, and some harmful organisms may increase in number when it comes to individuals having type 2 diabetes as compared to control. Now this is basically because there is an abnormal supply of glucose in the bloodstream. Okay, and obviously the retention of lot of glucose in the bloodstream, because the body cells are not able to take it up, leads to or creates a lot of imbalance in the kind of microorganisms that different parts of your body is going to possess. And that's why you would have also seen the diabetic people, they are so prone to getting infections. मतलब diabetes और blood pressure ये दो ऐसे conditions है कि एक बार इनका तुम्हारे जिंदगी में एंट्री हो गया ना, then it is like an invitation card for all other kinds of disorders and diseases that you can suffer. It is an open invitation, believe me. Even if you end up suffering with some kind of a condition and you need a surgery, a person who is diabetic and a person who is having blood pressure has to go through a tremendous kind of pressure, because until and unless your blood pressure and diabetes is brought to control, they will not even touch you. Do you know this? If you have to undergo even the simplest of the possible surgeries, and if you are diabetic, if you have blood pressure, they are not going to touch you. The reason for this is, students, the diabetic patients का जो healing rate होता है, the rate at which wound healing takes place in diabetic patients is extremely low. Do you know why? That is for a basic reason that their cells do not get enough glucose. And as they do not get enough glucose, they do not produce enough ATPs. And if they do not produce enough ATPs, cell division is not going to be possible. And if cell division is not going to be possible, wound healing is something which is going to be phenomenally slow. So you would, I don't know if you have heard of this, but it's very interesting, and I'm going to take time to tell you this, that In people who are diabetic, there have been instances wherein they have undergone a surgery, okay, and they have, got stitches, and you would be knowing that these days the stitches are done using biodegradable materials, okay, such that the stitch automatically dissolves there, and by the time it dissolves, the rate at which it, gets broken down, automatically your wound has healed. But in diabetic patients, the stitches fall loose by degradation, the wound has not healed, the cut opens up. Do you understand what I'm trying to say? Why is this taking place? Because the wound healing process is slow. It is much slower than the rate at which that biodegradable material is going to degrade. So this is a risk, right? And they are more prone to infections. So surgery means exposing internal body to all kinds of microorganisms that might be present. So it becomes difficult. In case of blood pressure, अगर एक गलती से भी नस को कुछ लग गया ना, so it is going to be like a fountain of blood that comes out. It will become very difficult to maintain the blood flow or stop the blood flow there. Because the pressure with which high blood pressure patients may, the pressure with which the blood flows is phenomenally high. There's going to be excessive blood loss and it could turn out to be fatal. The patient may lose their life. So it becomes very, very crucial to analyze that, does all this have roles to play, or rather does all this have connections with the microflora? Very important. Now, a relative increase in unnecessary kind of firmicutes, which are not the normal firmicutes which are found in the gut, but they are all useless ones, and decrease in the number of bacteroids have been seen in obese mice and in obese human subjects also. Okay, in some cases opposite condition has also been observed, which makes it more confusing. Microbiome में यही प्रॉब्लम है कि अगर मुझे एक ही टाइप का पैटर्न नहीं दिख रहा और मल्टीपल पैटर्न्स दिख रहे हैं, तो फिर उनका रोल एनालाइज करना और मुश्किल हो जाता है. तो ये सारे नाम याद रखना इज अ डिफिकल्ट टास्क, एंड आई डोंट एक्सपेक्ट आल्सो कि आप सारे के सारे नाम याद रखो, कहाँ पे क्या बढ़ता है. लेकिन जस्ट ट्राइंग टू अंडरस्टैंड कि इसका लिंक क्या होने वाला है आगे जब हम फंक्शन डिस्कस करेंगे, दैट बिकम्स वेरी सिग्निफिकेंट. तो ऑपोजिट चेंजेस भी रिपोर्ट किए गए हैं. कुछ लोगों में लैक्टोबैसिलस, बिफिडोबैक्टीरिया और मिथानोबैक्टीरिया के प्रपोर्शंस में बदलाव नोटिस किया गया. अब सबसे बड़ा सवाल आता है द चिकन एंड एग क्वेश्चन. अब तुम बोलोगे इसका क्या मतलब है? चिकन एंड एग क्वेश्चन मतलब जैसे हम पहले पूछते हैं कि मुर्गी पहले आया या अंडा पहले आया, वही क्वेश्चन यहाँ पर भी अप्लाई होता है कि ओबेसिटी के वजह से माइक्रोफ्लोरा चेंज हुआ है या माइक्रोफ्लोरा चेंज होने की वजह से ओबेसिटी का इशू हो रहा है. Do you understand what is the question over here? That is obesity the reason for change in microflora, or the change in microflora is the reason for obesity? This question has been there for an extremely long period of time amongst the research community. And there are a lot of US universities who have been investing their time and money in understanding the aspects of obesity, as to how obesity takes place, why does it take place, what is the reason behind it. Okay, but what is their exact role? This is what we will discuss tomorrow. Okay, I'm going to leave you a little early because I'm in college and I have to attend a meeting. So I'm going to stop at this point, but I want you to think a little more about it. That what do you think? Obesity is responsible for the change in microflora, or the microflora is responsible for obesity? So I will leave you at this point with this question, okay? And think a little about it, and tomorrow when we discuss in more details, we will try to understand that, what exactly happens when you talk about obesity. So do you have any questions till this point? Anyone?
No, sir.
No, sir.
Okay, so the test paper, the form for this particular section, I will post it give me some time, maybe by afternoon, I will post it on Wiki. So please see to it that you solve those questions by tomorrow's lecture. Is that okay with all of you?
Yes, sir.
Yes, sir.
Yes,
Yes,
sir.
sir.
Yes, sir.
Yes, sir.
Okay, great then. So I will post it on Wiki and I'll drop a message on WhatsApp group that the form is posted so that you don't have to constantly sit and check. Okay?
Yes, sir.
Yes, sir.
Okay. Fine then. See you all tomorrow. Thank you so much.